Hello boys, welcome back to another interesting video tutorials, and this session is meant for class 12th biology students, and this is for 29th August 2020. We have been discussing about gene expression both in eukaryotes as well as prokaryotes. Particularly in our last session, we were talking about how gene expresses itself in prokaryotes such as bacteria. Thanks to two scientists, Jacob and Monod, for projecting lac operon concept. Now we move on to another interesting topic in this chapter, that is Human Genome Project. It is abbreviated as HGP. It was launched in the year 1990. So US government started Human Genome Project in 1990, coordinated by Department of Energy and the National Institutes of Health. What is genome? The whole hereditary information of an organism that is encoded in the DNA is called genome. All the genes present together in the DNA, that is genome. So HGP is a mega project because the entire human species, irrespective of which country we belong to, which continent we belong to, the Indian genome, African genome, Asian genome, like that, all the human beings that are available in the world and their DNAs. You can imagine how long it is, how big the project is. Hence it is a mega project. Human genome have approximately 3 into 10 to the power 9 BP base pairs. So the cost of sequencing required is dollars per base pair, $3 per base pair. Then the total estimated cost of this project is so huge, $9 billion. You know, $1 is roughly 74 rupees now. The obtained sequences were to be stored in typed form in books. If each page of the book contained, let's say, 1000 letters, each book contained 1000 pages, then 3300 such books need to store information from single human cell. This is only single human cell. So always for any project there will be aims, goals. Okay. So what are the aims of this project? First of all, to identify the approximate 20,000 to 25,000 genes in the human DNA. So on an average, it is predicted that there will be 20,000 to 25,000 genes in human DNA. So we need to identify each and every gene of that 20,000 to 25,000. To determine the sequences of the 3 billion base pairs that make up human DNA. You know that bases will be the nitrogenous bases, 3 billion. And third, to store this information in databases. Fourth, to improve tools for data analysis. So what is that we get? Out of this, we get the data. And now we need to analyze the data so that we understand if the data is like this, what are the genetic diseases that a human being gets? If data is to be rectified, whether genetic disease can be rectified, characters, traits, everything can be analyzed. To improve tools for data analysis. To do this, we need some instruments, tools. So the aim of this project is to improve the tools for data analysis. Like we have got already biotechnology, and in that we have got various tools we will discuss in our next chapter. To address the ethical, legal and social issues that arise from genome research. Already there is a lot of discussion on what we call made up babies, or what we call syndrome babies. That is, all the good characters will be pumped into a zygote by making all good genes into that, and we can design the baby. So the concept is called designer baby. And then of course already people are having hue and cry. If all the citizens of the country are having same appearance, same characters, same behavior, how it would be. If you have variety, then only there is a lot of what we call social aspects, legal aspects. If all the citizens are of same type, same phenotype, same genotype, just imagine. So there is a lot of hue and cry about this. And what are the methodologies that we follow? The methods involved two major approaches. First one is identifying all genes that expressed as RNA, expressed sequence tags, we call it ESTs. So identify all the ESTs. Second, blind approach of simply sequencing the whole series of genome that contained all the coding and non-coding sequence. Later, assigning different regions in the sequence with functions. So it is called sequence annotation. That means we have seen in our last session mRNA, tRNA, rRNA complex. And we said the mRNA has got codons and the tRNA has got anticodons. So there was a matching of codons and anticodons, and when that particular codons and anticodons, triplet codes match, then particular amino acid was there. So now here, scientists wanted to play with that, with the sequence. They wanted to change the sequences. They wanted to change the sequence of the genes that are formed like that. So, and see what happens. Salient features of human genome. The human genome contains 3,164.7 million nucleotide bases. The average gene consists of 3,000 bases, but sizes vary. The largest known human gene being dystrophin has 2.4 million bases. The total number of genes is estimated at 30,000. It is lower than previous estimates of 80,000 to 1,40,000 genes. So to begin with, scientists were estimating that human DNA will have 80,000 to 1,40,000 DNAs. Now the total number of genes is estimated to be much lesser, around 30,000. Almost all nucleotide bases are exactly the same in all people. And look at that, 99.9% nucleotides will be same in all the human beings. The functions are unknown for over 50% of discovered genes. So we have discovered some genes, but we don't know what work they are, what specific character they are responsible for. Less than 2% of the genome codes for proteins. It is only less than 2%. So 246 million base pairs. Look at that. Has given varieties of pairs and what is responsible for that. Like you can see prostate cancer, brain cancer, so all these characters, dyslexia, these are all responsible. This is the wonderful discovery, including anemia, hemophilia. Look at that, almost all genetic diseases. So this is the greatness of this project. And 50 million base pairs. Short stature, homeobox, Y-linked. Short stature, Larivial disynchronizes. Langer mesophilic dysplasia. Interleukin 3 receptor, Y chromosome. Sex determining region in Y chromosome. Gonadal dysgenesis, XY type. See, so many. Male infertility due to spermatogenic failure. Everything is identified in even Y chromosome. So repeated sequences make up very large portion of the human genome. Chromosome 1 has most genes, 2968. Out of how many chromosomes we have got? 46. Out of 46, chromosome 1 has 2968, and the Y chromosome just now you saw has very few, 231 genes. Scientists have identified about 1.4 million locations where single base DNA differences, that is SNPs, single nucleotide polymorphism, we call it SNPs, single nucleotide polymorphisms. This information helps to finding chromosomal locations for disease associated sequences and tracing human history. That is the use of this. So these are all the uses of the human genome project. And This Human Genome Project was closely associated with the rapid development of a new area in biology called Bioinformatics. So when you go to PG level in the universities, you can go for MSc in Bioinformatics. The goal of the human genome is to develop a genetic linkage map of human genome by identifying thousands of genetic markers and mapping them in the genome. To obtain physical map of human genome by cloning genomic DNA into wax and cosmids to sequence the entire human genome. So two factors that made this possible are: number one, genetic engineering. We will be talking a lot about genetic engineering in biotechnology chapter. And genetic engineering techniques made it possible to isolate and clone any segment of DNA. Second, availability of simple and fast techniques for determining the sequence in the DNA. So mainly the methodology that we adopt is ESTs, expressed sequence tags. So remember this, ESTs, focused on identifying all the genes that are expressed as RNA. And second one, sequence annotation, sequence the whole set of genome that included all the coding and non-coding sequences, and later assigning functions to different regions in the sequences or the methods used to sequence human genome. So basically it is this human genome, and applications where can we apply this? The having a complete sequence of human genome will enable a radically new approach to biological research. That is, a systematic approach on much broader scale. All the genes in genome or all the transcripts in particular tissue, particular organ, tumor can be studied. It will be possible to understand how the enormous number of genes and proteins work together in interconnected network in the chemistry of life. So these are all that we can understand. And further, we'll be in our next session, we'll be studying about another very interesting one which helps us even in society, biology in social use. That is